Heredity. What is it? It's the passing of traits from parents to offspring. From parent to offspring. Now, it's important to note that all organisms use cell division to grow and reproduce. Now, in particular, the multicellular organisms use cell division for this growth, right? And maybe even repair. But unicellular organisms, however, use cell division literally solely for reproduction. So let's look at an example of a few of these traits. Okay, traits are like characteristics. So a characteristic of an organism can be inherited, like eye color, height, hairlines, but it can also be acquired too. We don't care about the ones that can be acquired, right? Like sun bleaching our hair or getting a tattoo. We're not talking about that. We're talking specifically about the ones that can be passed on from generation to generation. So I'll just put here can be inherited, like eye color, hairline, height, etc. Now these vary in different kinds of complexity. So with eye color, it's way simpler when trying to identify its probability from being passed on from one generation to the next compared to like height, which is a lot more polygenic, so a lot more variance is required. But we'll get to those calculations and creation of diagrams a little bit later on in the playlist. So for all this to happen, what is the unit of this inherited material? That is called the gene. So I'll put genes are the unit of this material. So let's look at earlobes. This one here you see on the bottom of the ear. You notice how it looks kind of deformed, right? Kind of like that. This is a detached earlobe. But this one here, you notice how they're all attached pretty linearly, right? So it feels like they're not, feels like there is no concave portion. It's just straight. So this is what we call an attached earlobe. Which one do you think is the most common? It's actually the detached. This is technically dominant. Most people have it. And we'll talk about dominant and recessives as well. But for now, just think of it as which one's the most common. All right, ability to roll your tongue. This one here is obviously able to roll. This person is not able to roll their tongue. Which one do you think is more common? Yes, it's actually this one. This one is more dominant. All right, so this is obviously a roll tongue. And this is obviously can't roll tongue. Hair types. This one is curly. This one is straight. Which one do you think is more dominant? More common? It's this side as well, okay? The left side, so this would be more dominant. Okay, so the curly hair is more dominant over the straight hair. Okay, when you clasp your hands together, which one do you think is more common? When you put your right thumb on top of your left thumb, or when you put your left thumb on top of your right thumb? It's this side again. When you put the right thumb on top, this is more dominant. Right, so I'll just put right over left, and this one is left over right. All right, this one's more common. Now we can go on and on and on and on, right? So dimples, Okay, what do you think about dimples? Do you think they're more common? Or do you think no dimples are more common? Dimples are actually dominant. Very common, actually. What about color blindness? Are the population of people who are colorblind more dominant, so more common, over the people who are not colorblind? Obviously not, right? So this one is obviously recessive. Most people are not colorblind. And you'll actually know that this is sex specific. And we're going to learn about why males have a higher chance of being colorblind as opposed to females. So we don't need to make more examples of traits. I think you get the point. All of these are inheritable. And so it's very important to understand genetics, the field of biology that studies how genetic information, these traits that we've just been talking about, how are they passed from one generation to the next? Well, to understand genetics, you need to understand where this genetic information comes from. Of course, genetic info comes from DNA. This should be pretty common knowledge, right? So DNA is also known as deoxyribonucleic acid. And each DNA molecule is made up of hundreds of thousands of chemical subunits that make up the instructions. All right, that instruct the cell. So all hereditary material is stored in the DNA. All of the organisms, whether it's bacteria, plants, animals, fungi, protists, they all require DNA. All right, and what are they composed of? They're composed of two long polymers. All right, so these polymer chains are repeating units. As we mentioned up here. Okay, so what are the names of these subunits that we have repeating in a pattern-like fashion? They're called nucleotides. So these are the building blocks of nucleic acids, what we learned about in the macromolecules video. Okay, remember amino acids make up protein, monosaccharides make up disaccharides, disaccharides make up polysaccharides for the sugars, glycerols and fatty acids make up a bigger fat molecule like a triglyceride. Nucleotides make up nucleic acids. In particular, that we're going to be focusing on right now is the deoxyribonucleic acid. So deoxy means no oxygen. I mean, there's obviously an oxygen. So I want you to think of this as one less oxygen, not no oxygen, compared to like a ribonucleic acid. Okay, so if we were to compare this with a ribonucleic acid, this ribonucleic acid has an extra oxygen. Deoxy means one less. That's how I want you to think of it. All right, and who discovered this DNA? Technically, Watson and Crick won the Nobel Prize of publishing their work that the DNA is made up of a double helix in 1953. However, it's important to note that they stole this work from a predecessor, particularly from someone named Rosalind Franklin, who worked under Wilkins. So Rosalind Franklin captured this image using X-ray crystallography. You see you have that spiral shape, right? Okay, you see that? How it's in an X spiral-like shape. This is known as photo 51. Again, using that method of X-ray crystallography. So credit really should go to Rosalind Franklin for discovering how DNA looks like. And it's just unfortunate that she was not recognized back then for the work that she did. Now looking at this DNA structure, let's dive into it a little bit more with a little bit more detail. It consists of two strands, all right? So you see how this strand over here is one strand, and this strand over here is another. And you see how they're going in opposite directions. How are they attached together though? Well, you see how in the middle there, all of these ones, they are strongly bonded, actually through hydrogen bonds. And you should be a little bit familiar with hydrogen bonding based on what we've learned about in electronegativity. So there's a bond between these bases. You see that A and you see that T and you see that C with that G. These are the bases. So nitrogenous bases, right? And A must be paired with T's and C's must be paired with G's, all right, as you can see there as well. Now, what is the actual backbone though that is keeping it all together? It will be the phosphate group. As you can see over there, there's a phosphate on it. And you should also know how to draw this phosphate, because we looked at it in a previous video. Right, you should be able to know how to do this as a Lewis structural diagram. All right, so this backbone is made up of alternating phosphate groups. All right, so phosphate groups. But there's also one more group that helps bind all these together. Right, it's not just the phosphate groups, it's also the sugars. So you see these sugars over here that are on the inside, in the middle, in between these phosphate groups and the nitrogenous bases. A particular sugar, like we've already said, for DNA is deoxyribose, not ribose. All right, so let's bring this down here. This would be a sugar. So if we zoom into here, you see how the ribose has an OH here on the bottom, whereas the deoxyribose sugar only has an H over there. Okay, so it has one more oxygen compared to this one, like we said earlier on in this video, okay? So because this has one less oxygen, we call it deoxyribose. So if I bring them back together here, okay, so we're looking again at the building block of DNA, the deoxyribonucleic acid. Again, what makes up nucleic acids? Nucleotides. So this here is a nucleotide. You bring these together long enough, you get a nucleic acid. And that sugar over there that we looked at, that's what we see over here. So we've already mentioned that there are three parts. We have that nitrogen base, that's over here, that's one. We have the sugar, that's this one over here, so that's number two. And the sugars differ depending on the type of nucleic acid. So if these nucleotides get together, but in particular use this kind of sugar, right, the deoxyribose sugar, it would be a deoxyribonucleic acid. But if the nucleotides here got together and had their sugar subunits using a ribose sugar, you would get a ribonucleic acid. And then lastly, either one, the DNA or the RNA, they would also need a phosphate group. And why do these strands separate away from one another? It's because these phosphate groups, as you can see, are negatively charged. In fact, the Lewis structural diagram of this phosphate group actually has negative symbols over here. It's missing one over there, it's missing one over there, okay? But there should be about three, because one of these are actually double bonded, so there should actually be a double bond over there. All right, so these single bonded ones should have a negative charge. See the video link in the description if you have no idea what I'm talking about. All right, so because it's so negatively charged, these negative charges want to separate away from these negative charges because electrons repel. There's a lot of repulsion. So this repulsive force from all these phosphate groups, or phosphates with these phosphates, they do not like each other. Hence it bends them in such a helix-like fashion. Now these two strands are actually anti-parallel. All right, so the two strands are anti-parallel. What does this mean? So the direction of polarity runs from a 5' end to the 3' end. So I won't be too technical here, but I just want you to just zoom in here. You see that sugar that we looked at earlier, right? You see what's branching off of that sugar? The phosphate, because remember, they both form the backbone of this whole structure, the rungs, right? This ladder-like structure. So that structural integrity is thanks to these phosphates and then the sugar. The phosphates and the sugar. So if we look at this phosphate over here, if we were to deem this as a 5' phosphate, then the complete opposite end of that sugar won't have the phosphate end. Okay, so again, if the phosphate group here is on the top, it's at the very, very top of this sugar, what's going to be at the very bottom of the sugar down over here? So let's draw the sugar here. See how the phosphate is on top, right? This was that phosphate group. So the 5' phosphate. What's going to be on the very bottom? Instead of a phosphate group, what is hanging off is actually a hydroxyl group, right? The OH. Now we call this the 3' end. Okay, so the 3' end hydroxyl. So if that's the order for this particular strand over there, again, the very, very top end of that sugar has the 5' phosphate, but the very bottom end of this sugar is a 3' hydroxyl. How about the exact opposite strand? It's going to be like vice versa. So if this was, again, the sugar, at the very bottom here, it's not going to be aligned with the other strand's hydroxyl group. Remember, hydroxyl groups are a 3' end. We have to do the vice versa. So we're going to do the opposite, the 5' end. Okay, so it's going to be 5' end over here. But which one is the 5'? It's the phosphate group. All right, so this would be the phosphate. All right, so I'll just put a PO4 3- over here. That's the phosphate. So we just continue this up all the way to the end. You saw there's that phosphate on the bottom there. That means there must have been a sugar here too. Okay, so if we were to draw that sugar there, again, the phosphate's at the bottom end, so what's on the very, very top end? It has to be the 3' hydroxyl. So phosphate is at the very bottom here, at the opposite end, and then we come over here, at the very, very top end, you have the 3' hydroxyl. Again, same thing, we go from 5' to the 3'. Similar to what we just looked at before. From the 5' to the 3'. Alright, it's important for you to know that these strands run antiparallel. So let's look at nitrogenous bases with a little bit more detail. A T is known as thymine, and a C is known as a cytosine. A's are known as adenines, and G's are known as guanines. Remember, A's and T's go together, C's and G's go together. So I'll just put here AT goes with GC, remember that, okay? AT with GC. So the ones on the very left side over here, I want you to categorize them as very similar, as more similar structures than these guys. So these here are known as the purines. So purines. Whereas this side over here, these are known as the pyrimidines. I think this might be with an I. So anyways, what does this mean? It means that purines complement pyrimidines, and pyrimidines complements purines, all right? So A's complement T's, G's complement C's, and they form those hydrogen bonds in between, all right? So again, this one and this one are hydrogen bonds. And it's important that they're strongly connected with a lot of polarity. Otherwise, what if it's easier to break them apart? Isn't that really problematic for your DNA? You don't want your DNA to be broken apart, forming new bonds that it shouldn't be doing. This can result in serious health consequences, diseases, or even death. So we want it to be nice and strong. Okay, and it's not just single bonds, all right? This one here, actually, there's about 2 bonds happening. And the G's and the C's actually have 3 bonds happening, okay, the hydrogen bonds. Now, you can make lifestyle choices to ensure that you reduce the risk of these breaking apart. For example, staying away from radiation, so not having too many X-ray scans per year, or not staying out in the sun for too long and getting burned, avoiding places that have an abundant amount of chemical toxins, washing your hands, wearing a mask, to ensure that you don't get infected by bacteria and viruses so often, because all those things can result in mutations or errors when we replicate, because all these things are part of DNA and DNA's got to replicate. This is why you're made up of 100 trillion cells, because every single cell has a DNA that makes up who you are, and they replicate it. They make more and more and more and more and more. That's the reason why you're so big today compared to when you were a baby, because thanks to those replications, you became a bigger and bigger and bigger cluster of these cells. And as you are growing and repairing yourself, you want to ensure that we minimize the amount of chance that errors occur. But again, sometimes it's unavoidable, right? Like bacteria and viruses, you're always going to get sick. Hard to avoid it, but just do the best that you can. All right, so I'll just put up here, incorrect pairings can result in mutations. All right, so do what you can with lifestyle choices. All right, so lifestyle choices, all right, can reduce chance, but natural occurrences do happen, which is unavoidable. And this is just unfortunate, but that's just the way life is. It's not fair. All right, so scientists have discovered that if the sequence of the strand of DNA is known, we can easily find the other strand. So let's say, for example, we got ATG and GCT. What do you think the other strand's nitrogen base is going to be to complement it? Remember, A goes with T, so this will be a T. This will be an A. This G, what is it with? It's with a C. What is this G with? Again, with a C. What is the C with? With a G. And what is this T with? It's up there, it's with an A. So this will be the complementary strand of bases. All right, so packaging DNA. So DNA exists as chromatin. And they're kind of like spaghetti looking like strands of its genetic material and some protein. Okay, so there's like some protein histones as well. But DNA has to eventually mature and become a shape looking more like this, as opposed to kind of like what I just said, like spaghetti strings. So this organization that it eventually becomes is thanks to some of the proteins, also known as some histone proteins. All right, so the histone proteins are where these things, the string-like chromatin, that is pretty much what DNA is, right? It wraps around these histone proteins. All right, so organization of DNA prevents it from tangling thanks to the histone proteins. All right, so it keeps it in good shape. And eventually when they're done wrapping around with one another, they eventually start looking like this. Now this is kind of an advanced stage, okay? This is not when it's resting. This is literally when it's about to start dividing. This is going to be in the first phase known as prophase. So when it's about to divide, these chromatins are kind of divided in two, okay? You see how it says sister chromatids, right? So this is one sister, this is another sister chromatin. This will eventually split right down the middle at here, okay, the centromere. Now, why did it do that? Because in the beginning, remember how I talked about these spaghetti-like strings? Okay, technically that was all just this stuff, for example. Okay, all of these are this kind of structure. But like I said before, DNA needs to replicate. It's what makes you who you are. The reason why you're able to make more and more cells is because of replication. So what we had here in the beginning, replicated. So this end replicated to this end. So if you split this apart, it's no problem. Because if this goes and starts dividing to make more and more and more cells, and this goes away to start dividing to make more cells, that's fine because they're exactly the same. Okay, at the very beginning, this replicated to this anyways. This is why every single cell in your body has exact same DNA to one another. All your skin cells are the same. All your muscle cells are the same. All your nerve cells are the same. They're all genetically the same. Now, we just can't divide forever, right? Eventually, the ends of these chromatins start to shave off a little by 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 a little. This is what happens when you age. Every time you divide your cells again and again, a little bit of this part of your DNA starts to shave off. Now, we're not going to get into the specifics of introns and exons. That's for another video. I just want you to know that there are sequences of code over here that eventually get shaved off every time it divides. This is why you don't age continuously. You lose so many of these telomeres that it slowly ends up killing you. This is why you can't live forever. All right. So I also just want you to know that this whole thing right now, when it's done that whole replication from this to this and maybe this to this, and you have a pair of these sister chromatins, okay? But because they are sisters to now to one another, we actually change the ending, right? The suffix to IDS, so we call them sister chromatids. But obviously, eventually, when this splits off in half, right, this will now be off in its own direction, and this will be off in its own direction. Then they would be like chromatins again, right? But what's odd is, again, this is just a language thing, but then this chromatid, right, this sister chromatid, once it's broken off, it's now a chromatin again, but we actually call it a chromosome. Why is that a little bit weird? Because right now, when they're all stuck together, as you can see over here, this is actually an official chromosome. So a lot of students get confused when this breaks apart and then this kind of sister chromatid breaks off from its other sister and is a chromatin again. Why they call it a chromosome in literature. It's just something you have to know. When the mitosis is complete, when the division is complete, just know that this chromatin is now called a chromosome technically. But it's also a legit chromosome at the very, very beginning when everything's replicated and is ready to divide. Okay, so this X-looking shape of DNA, this is called an official chromosome too. Alright, so chromosomes also vary in size and shape. Now, most multicellular organisms have less than 100 chromosomes in the nucleus of each cell. Okay, so if I have a cell here, okay, let's say this is a human. How many chromosomes are in each cell? Well, for humans, there's 46. All right, so we have 23 pairs of chromosomes, but all together we have 46 chromosomes. Okay, 46 of these guys. But in, let's say, a mosquito, they only have 6, right? 6 chromosomes. Regardless, all of these numbers that you see over there represents literally all of the DNA that makes up who you are, or makes up the mosquito. All right, so for this mosquito, again, it was 6, right? So all 6 of these are the collective full amounts of DNA that makes up the mosquito and how it behaves and how it looks. All right, so it's very important for you to understand that these chromosomes that you see and that we're going to be talking about often, they are literally the DNA. These are all codes of DNA. All right, so for the cell cycle, it has 2 stages. All right, the growing stage and the division stage. And you should be familiar with this. Okay, this kind of sounds like mitosis, right? Because it is. All right, aka mitosis. All right, so in the very, very first step, okay, even technically before mitosis, there's a preliminary step known as interphase. Once interphase finishes, then mitosis occurs. But this interphase is that growing phase. Growing occurs. All right, so the genetic material is not a chromosome. The genetic material is the chromatin. Remember that spaghetti-like fashion? Okay, you just have strings around. Okay, these are all just chromatins. All right, this is all happening in the interphase. All right, so as a cell then prepares to divide, so to go into mitosis, the chromatin thickens and shortens. Okay, and with the help of those histone proteins, they become a nice and X-like helix-shaped structure. Okay, and we now call this an official chromosome now. And don't forget, they also have to replicate first so that when they divide, they don't lose any information. They don't lose half their information, right? You want to make sure that they divide first so that when you split them apart, you're still good. You still have 100% of you left. All right, so let's pretend this is just one chromosome, okay? This one is obviously unduplicated. I'll just put here unduplicated. But then when it starts to duplicate, it maybe kind of looks like that. All right, this one here is duplicated. All right, so this chromatin here, that's this one, okay? I'll do it in here. Okay, this was that chromatin. But it's gonna do its little replication, so now it's a duplicate of one another. All right, so this red one here, this is the sister chromatid to that former previous chromatin. They're connected by that centromere over there. Right here, I'm just gonna label this properly. Okay, so that the little dot right there is the centromere. This was that chromatin. Right, same with this one. This is that same chromatin. And this one here is the new sister chromatid. And eventually they will split. Okay, this one is going to split off, and this other strand is going to split off too. Now again, it is odd. This is a language thing, right? I know we've been looking at this as the chromatin. Some researchers, some scientists may call this a chromatid as well, but it's more commonly known as a chromatin. All right, we only use a chromatid when the sister to the former chromatin has been made. Okay, that's when we generally use a chromatid, a TID, when there's a pair now after replication. So what is a gene? All right, a gene is a segment of that DNA, so a particular area that carries information that helps to produce a particular trait. All right, so each gene occupies a specific location. This is known as a locus. All right, so each gene occupies a specific place. So again, if this is your DNA here, you have specific places, like these bands that you see here. Okay, we'll just look at this particular band here, and maybe that one. Okay, this is a particular loci of a gene, right? That's plural for locus. So let's say this particular area dictates the color of a petal on flowers. So whether it's purple or pink. All right, this one may be purple, this one may be pink, but they're in a specific area to code for that particular trait, color of the petal. If we were to get another one over here and mix them together, it's possible that one parent's purple color petal might be matched with the pink over here, also within the same specific loci area, because this area here is coding for the trait of color of the petal. These different colors are different versions, right? So these different versions of a gene are called an allele. So I'll put it over here. An allele is a different version of a gene. So this one parent, when met with this parent's and fertilization occurs. This one here was the allele for a purple petal flower, and this one here was an allele for a pink petal flower. They will be homologous, so they will be paired together, but we will get more into depth of this when we get to meiosis, when genetic variation occurs. So we're still within the realm of mitosis, so I don't want to jump too much into meiosis, but I just really want you to know that there are sections of a gene that code for a particular trait, and there can be different versions of this particular trait. These are alleles, because a trait for a petal of flower can look different. This one can be purple, this one can be pink. Or what if they're both pink? That means for sure the next generation is probably going to have a very, very, very high likelihood of it being pink as well. But again, before we get too carried away to sexual reproduction, let's just jump back to asexual reproduction. All right, the mitosis that we're covering. All right, a new individual is produced. Okay, but this is from a single parent by cell division. So there's no sex happening. No sex cells were involved at all. All right, so that chromosome literally just duplicated on its own. Okay, and then it divided. All right, so that each new cell, all right, we call this a daughter cell. All right, a new cell that is formed, we call it a daughter. So each new cell that it's split apart into receives one exact copy of each chromosome. Okay, so then this one is obviously identical. It is completely the same. All right, so it has an advantage. Okay, what's the pro to this? All right, there's no need to seek a mate. So no specialization of mating behavior is required. So it's very efficient in terms of energy. But there is a con. If a disease breaks out, since everyone is genetically identical and they're genetically susceptible to be harmfully impacted from that disease, then everybody dies because no one is different to maybe have a variant to be resistant to it. So there's a risk of eradication, right? But overall, in the beginning, all cells must be derived from pre-existing cells. We all started from one cell. All right, this is why mitosis is very important. And again, these occurs only in somatic cells. All right, somatic just means body. Okay, so every single cell that is not a sex cell. All right, so cell division consists of that resting period of interphase. All right, but it's not just resting here, it is getting ready for the mitosis section. Okay, so a lot of replication happens here. So replication occurs. There is growth. There is repair. There is a checkpoint as well, right? It checks to ensure that it's actually safe to start mitosis. All right, then it gets into mitosis, right? This is the actual division. This is the division. Because once you start dividing, you want to make sure that everything's good, right? Everything's replicated well, everything's grown well, everything has been repaired to ensure that the division goes smoothly, that if any cell destruction needs to happen so that healthy cells are made and ready to go. That's what happens at the checkpoint, right? Because if the cells aren't healthy and they start going into mitosis, then these are going to start dividing, but they're all now dividing from the messed up cells created before that were not double checked properly. This is why this takes a long time. It's got to be very meticulous in its approach. The division is actually very quick. All right, so when we get that genetic information and divide it, we want to divide it equally into two new nuclei. All right, so I'll just put this down here, the point of mitosis. To divide genetic material equally into two new nuclei. Right, because the DNA lives in the nucleus, right? All right, so what's happening inside the nucleus? First phase was G1, right? This is just rapid growth and metabolic activity. All right, this S phase is now that replication phase, all right, of that DNA, all right? And then the last phase of that interphase is when the organelles start replicating, right? Like the centrioles, the mitochondria, etc. All right, so the cell prepares for division. All right, and once that's all good, then we move on to the first stage of mitosis. All right, so this is the first stage of mitosis. All right, so let's start dividing now. All right, so you see these centrioles, they start moving to the opposite side. So as this starts going towards here, this starts going towards here. Once they plant here and plant here, they will start attaching onto the chromosomes and here in the middle. You notice how the chromatin have condensed, right? So now they're very, very mature looking chromosomes, tightly bounded. This envelope starts to slowly break apart and will become no longer visible shortly. The spindle fibers start coming out of these centrioles and they'll start attaching onto these centromeres, remember? Why? Because again, they gotta split off in half, split off in half, split off in half, all right? One side is going to go one direction, the other one is going to go in the other direction, into their new respective outer cells. All right, so the chromatin condenses, nuclear envelope, so Ne breaks down, so they can actually split apart without a barrier in the way. All right, and the spindle fibers start to attach to the centromeres, all right, of these chromosomes here in the middle. All right, so now these chromosomes are now lined up here right in the middle, right? And it's known as the equatorial plate, okay? That imaginary line that's in the middle there. So I'll just say chromosomes align at the equator. All right, at this point it's very, very apparent that the spindle fibers have attached to its respective centromere. All right, so spindle fibers are now ready to pull. All right, at the centromere. And of course, you cannot even see that nuclear envelope. It is completely gone. Now they start to contract the spindle fibers and pulls towards the centrosome, okay, which has the centrioles. Same thing on this side. So spindle fibers contract. So they shorten and pull. All right, so now that the chromosomes have now split apart, what do they have? What have they become? They become two identical chromatids. Okay, because they remember they were sister chromatids, right? So then now these chromatids look very similar to those chromatins at the very beginning, okay? But remember, technically we can actually call this now a new chromosome though. Obviously different than the actual original chromosome. All right, now they move to those poles, right? Opposite poles, right? So these ones are at this pole at the top, right at the North Pole kind of area, and then these ones go down to the South Pole kind of area. And what's very obvious happening here is that nuclear envelope, it starts to come back. So nuclear envelope, the Ne came back, reappears. All right, so now those new chromosomes, right, or quote-unquote chromosomes, become thin again and more spaghetti-like. And the spindle fibers, I'll just put SF. Okay, the spindle fibers disappear, they're gone. All right, and here the division has complete. You see how a cleavage furrow has started to be formed? Okay, concaves inwards. This is the beginning of cytokinesis. Now, whether that's part of telophase, like a late part of telophase, or if it's just on its own, that is the end point of mitosis, doesn't matter. There's a gray area, right? It's a continuum, we don't know. But regardless, this whole cell membrane here, right, on the outside, pinches to create two official daughter cells. Okay, because unless this is actually broken, you don't actually have its own cell. This one here is a nuclear envelope. This here also is a nuclear envelope, but it's not its official cell. A cell has got to have all these organelles and everything, right? Centriole, the mitochondria, the lysosome there, okay, it's got to have everything. The cytoplasm on the outside. So we got to really wait until that thing splits in half. All right, and now you have the official identical daughter cells. This is one, this is another. So this is the official separation. The separation of that cytoplasm, and you see official division with two new daughter cells.